Thank you very much. I'd like to thank the organisers for giving me the opportunity to speak about one of my favourite topics, which is precision medicine, and particularly its applications in neuroscience, which has traditionally been one of those areas that's incredibly difficult for traditional genetic approaches to be able to address. We started PrecisionLife at a point where it was obvious that the Human Genome Project and other technologies were generating large sets of data that were going to be relevant to making decisions about new drug products that could be developed, or the matching of drugs to patient subgroups. But we didn't really have the analytic capabilities to do that. And that's limited the success of the Human Genome Project and its relevance across a wide range of more complex chronic diseases, of which psychiatric disease, neurodegeneration, and other neurological conditions are absolutely central. And that's because these diseases work in a very different way from some of the diseases that were known to us to have a genetic component when we went through the first round of the Human Genome Project. Instead of single base pair changes, for example, in sickle cell anemia, where you have a glutamic acid mutated to a valine, which creates a sticky patch on one of the subunits of hemoglobin, which sticks molecules of hemoglobin together into fibrils that sickle the cell and reduce its oxygen transmission capabilities. That's an incredibly simple one change, a change in structure, a change in function, and therefore a disease process. We kind of had an expectation that might be the same across a broad range of diseases. Of course, neuroscience has demonstrated that that's not the case. If you look at the results for a genome-wide association study in something like schizophrenia, you see that the GWAS SNPs explain only about 5% of the disease. And that's because fundamentally these are complex diseases. There are multiple genes interacting with other clinical and epidemiological and environmental factors coming together to exert a particular impact on the disease phenotype. And that coming together works across interacting networks of genetic and metabolic pathways that have nonlinear impacts on each other. And that's really why PrecisionLife exists. We found a way to exploit the signal for those interactions that are absolutely fundamental to complex diseases. So what do? Well, we take large-scale patient data sets. This, as I say, can include genomics, clinical information, transcriptomic, biomedical imaging, epidemiology, and environmental data. And we have a data-driven, hypothesis-free way of using that information to get under the skin of a single diagnosis. So this is a patient stratification of Alzheimer's disease. And you can see the colors represent different subgroups of patients. And the tiny little dots represent SNPs that are relevant to that subgroup, that patient subgroup's form of the disease. And that changes our view of the disease from being a single diagnostic label into a series of subpopulations, each of which has a slightly different disease etiology, each of which will therefore benefit from a different drug targeting a different mechanism of action. And I'll show you that in the subsequent slides. But what this allows us to do is obviously understand all of the different opportunities for creating innovative approaches to targeting some of the disease processes associated with these really difficult to treat diseases. So we can find novel targets and leads. We can generate patient stratification biomarkers, which can be used as inclusion or exclusion criteria for clinical trials, simplifying the clinical design by allowing the recruitment of a much smaller population, a much more targeted population, and having a faster readout of those clinical trials. And also, those patient stratification biomarkers can become complementary diagnostics. So knowing that a patient belongs to a green group instead of a red group and therefore has a particular mechanism at work allows us to be more selective about the therapy that we're going to prescribe for them. And that's of huge interest when you're putting a new drug on the market and working inside an outcomes-based remuneration model, being able to identify those and give tools to clinicians that support the use of your medication. Of course, there are other opportunities in predicting and preventing chronic diseases, identifying patients in the prodromal phase while interventions can still be deployed to them. But I'm predominantly going to talk about the drug discovery and indication extension opportunities in this talk. So rather like the Alzheimer's example that I showed earlier, this is an example in ALS. And you can see the clinically relevant subgroups of patients are the big clusters, and the SNPs associated with their form of disease are the tiny little dots. And this comes from our analytics and high-resolution patient stratification capabilities. But once we have this stratification, we can map those SNPs to the genes that they code for, those genes to metabolic pathways, and then we can also lay on all of the known active chemistry that works against those targets. And this is a bit of a treasure map now. What we can use this for is selecting novel targets which have high clinical relevance, which have good tractability for a particular modality, and ideally where we've got some tool compounds that are known that we can put very quickly into patient-derived cellular assays. That's what we did with ALS. We identified 33 novel genes in a whole range of different cellular mechanism categories. We chose 9 of the novel targets. We put them into a patient-derived cellular assay, which was a mixed cell assay of motor neurons, microglia, and astrocytes. And of those, 5, and each one of these targeting a different novel mechanism, 5 showed potential for improved motor neuron survival, some very, very strongly. So this is kind of interesting. It also allows us, as well as that innovation, it also allows us to start targeting some of those innovations at particular patient subgroups. And this is some further work that we did in collaboration with King's College, looking at clustering, stratifying the ALS patients on the basis of their underlying genetics, not just the Mendelian genes associated with different proteinopathies, but actually with some of the other more subtle mechanisms. And you can see that we have different patient subgroups associated with different genders, different rates of progression, different secondary diagnoses, and critically, different genes. So this starts to build a picture of disease. And I want to illustrate that further by going back to the Alzheimer's example. So this was data derived from UK Biobank. We took 900 Alzheimer's patients, 1,800 controls, the oldest population available without neurodegenerative disorders, and took their genotype data. When you run that through a standard analysis like GWAS, you find one hit. That's the ApoE4 locus. But running that same data set using our approach, we found 267 loci, mapping to over 100 genes. And this allowed us then to cluster those SNPs by the patients in which they co-occur to give us this high-resolution patient stratification view, a disease architecture, as we call it. And you can see that, yes, lipoprotein metabolism was the most important aspect of this, but we also saw signal associated with neuroinflammation, the regulation of monoamine oxidases, and neurotoxicity signaling cascades as well. So starting to understand the drivers of disease for different patients. And this becomes really interesting. What we wanted to do, we realized that these communities, these clusters, in many cases were oriented around genes that existed in the same pathways. So there was a real mechanistic drive across them. And we wanted to explore that in a little bit more detail. And we did. We clustered those further to identify 6 major patient subgroups, each of which had a different mechanism of action. And you can see here that, yes, lipoprotein metabolism is important for about a third of the patients. But there are other groups here as well. There is a group for whom prior viral infection causes changes in the immune system, which result in sensitization to neurodegeneration. There's another group for whom dysregulated autophagy and inability to maintain neuromuscular junctions is the primary driving feature. Another group for whom dysregulated metal ion homeostasis, particularly copper, is most important. Another group whose disease is driven by metabolic syndrome. And finally, a group who has dysregulated neurotransmitters, particularly serotonergic. Now, if I go back to the top here, you can see something striking immediately. There have been about 100 failed clinical trials in Alzheimer's disease, and they're all chasing the same basic mechanism. But that mechanism across the population we studied was only driving disease in about a third of the patients. And so even with the best drug aimed at that mechanism, if you choose your patients across all of these different subgroups, you're likely only to be able to demonstrate 32% efficacy, which is not enough to put a new drug on market. So you have to start thinking about using this in order to de-risk the choices that you're making, and also to make different choices. You know, there are druggable candidates inside each of these different patient subgroups. And in fact, it may be that an individual patient has more than one of these features, and that might represent an opportunity to do combination therapies. But whatever, we have patient stratification biomarkers that can delineate each of these patient populations. And that becomes an incredibly powerful tool for the onward development of new drug programs. A very good example is this serotonin dysregulation group, which you can see highlighted in black here. And if we were to target that, we would really want to take a patient stratification biomarker to allow us to define inclusion criteria that stacked the deck of a clinical trial with potential responders, and in that way be able to move the medicine through clinical development much more quickly and with a lot more certainty. So I've shown a lot of examples in a detailed stratification of neurological diseases. We've actually done about 35 disease studies. So this is a very scalable kind of view of disease. And you can see in neuroscience, we span both neuropsychiatric and also neurodegenerative and some other CNS disorders as well. And this is kind of interesting. It gives you an opportunity to do a number of different things. Firstly, you can see common drivers of those diseases. So we see the same genes turning up in schizophrenia and ALS and Alzheimer's. In fact, also through COVID and long COVID, identifying from the other direction those post-viral changes that lead to neurodegeneration and potentially to cognitive decline, possibly COVID fog. So you're starting to understand the relevance of mechanisms more generally. And this, again, offers another opportunity. Once you've done 35 different diseases, you can start to think about the relevance of that mechanism across secondary and tertiary indications. So in other words, we can take a drug that's on market or in development that targets a particular gene, and we can look for the relevance of that gene inside a secondary indication. And critically, this is bounded by patient stratification biomarkers. We can decide whether we're going to be able to identify potential responders at a clinically relevant, i.e. prevalent enough frequency inside that secondary indication that this might represent an indication extension or a drug repositioning opportunity. And that would be bounded, of course, by the patient stratification biomarkers that we're using here. So we've actually automated this process, and we can now do this at an industry pipeline level. So we can take all of the development and on-market compounds, even withdrawn compounds, and we can, in an automatic fashion, identify their relevance across different indications, across these 35 diseases that we've studied, and give a great deal of information about the patient stratification biomarkers, the potential to demonstrate efficacy, the credibility of the mechanism of action, and then start getting into other criteria like the dosage and route of administration, potential tox issues, freedom to operate, the market potential, and decide whether there are opportunities that we really want to go after. And this is what we have done. So across all of the industry pipeline, we've taken all of those compounds. As I say, we have tested them against a wide range of evaluation criteria beyond just the relevance of the mechanism of action and the patient stratification biomarkers. And we've got to the point where we can generate data packages supporting patent filings, method of use filings, linking the composition of matter for drugs that have existing patent life, typically over 5 years, to a new indication via the patient stratification biomarkers that describe the patients who are likely to benefit in that secondary indication from a drug targeting that particular mechanism. And this has been very successful. We've done this, we've validated this in a number of cases. One that I can talk about is dutasteride in severe COVID-19. Right at the beginning of the pandemic, we generated a preprint of the paper, I should say, looking at the factors driving severe COVID-19. And as part of that, we identified 68 genes. 48 of those genes have been now validated by the wider community. We identified 29 high priority repurposing candidates, 13 of which are now in clinical trials. One's just read out from phase 2 and demonstrated reductions in viral shedding and inflammation. So this was an example that we suggested, and you can see that there is significant supporting evidence now that in fact this compound dutasteride is actually active in and useful for severe COVID-19 patients, particularly in males. But there are many other opportunities. When we analyze the whole pipeline, and this is as of March 2022, and this is all of the public pipeline for companies worth more than $1 billion, as reported by Global Data. You can see that we have something in excess of 480 potential opportunities. And when we go and validate them against the criteria that I described earlier, that ends up being a list of about 180 opportunities. And 66 of those are in CNS disorders. And you can see that there is a range of prevalences for these across a variety of different development phases. I've blanked out the company names here just for a little bit of additional security. But each one of these represents an opportunity, not only for the inventor of that medicine to create new commercial opportunities for itself, but also for patients to have better options delivered quicker and more cheaply to market that meet their unmet medical needs. And finally, that's really what our mission is about. It's about understanding disease biology and the relevance of specific targets and mechanisms to different patient subgroups, generating those unique disease insights, that understanding of biology, and then using it throughout the ecosystem. So yes, lots of novel targets and disease programs, but also these indication opportunities and the biomarkers to support them and their use in clinical practice, and then even out into helping patients understand their own diseases. So I hope this has been very helpful, and I thank you for your attention.